So today I'll present our work on using focus ultrasound BBB disruption to improve the delivery of AAV vectors to the brain for the treatment of Huntington's disease. So right now there's a lot of justifiable enthusiasm around using gene therapies for the treatment of many neurological disorders. And much of this process stems from great work that's been done over the last 2 decades of identifying and characterizing the underlying genetic mutations that cause these diseases, and then developing an array of therapies that can target these genes. And there's now a large number of companies that have entered the space to try to translate these therapies into patient use. And there's currently over 100 ongoing clinical trials in this area, and a most recent success of a couple years ago of an approval for the gene therapy for spinal muscular atrophy. However, it's also clear that several challenges remain, as evidenced by the fact that several clinical trials were recently stopped, either for lack of efficacy or for excessive toxicity, including several patient deaths. And despite all these resources that are being poured into the problem, there is still only one therapy that's been approved for an application in the central nervous system. And likely one of the biggest obstacles that still remains is the challenge of delivering the therapy to the brain. So we think that focused ultrasound can come in and help here, and potentially has a number of unique advantages over existing routes of administration that could possibly reduce systemic dose, better reach deep brain structures, use repeated treatments, or improve coverage. So in this project, we want to validate that we can use focused ultrasound specifically for delivering an AAV9 vector and targeting Huntington's disease. So the experimental design is shown here, where we're targeting BBB disruption in one hemisphere, and then intravenously injecting an AAV9 capsid, surviving the mice for 3 weeks to be followed by histology. And the AAV vector that we're using carries both a microRNA and a green fluorescent protein. And for our purposes, even though the microRNA is targeting that Huntington's gene, we're just using the GFP to measure distribution. So the first study that we'll show is getting a validation baseline in just wild-type mice, where we do BBB opening and then look at both safety markers, measures of inflammation, and then also distribution of the green fluorescent protein and cell type specificity. And then in the second study, we're doing AAV9 delivery in 4 different groups that include 3 groups of Huntington's disease model mice, 1 strain at 2 different ages, and then a second strain at the same 2-month time point. And we've done contrast MRI to quantify the BBB disruption in these different groups of mice, and are about to start on the histology to determine GFP distribution. So here's the first set of results from the first study in wild-type mice. We're showing the 3 mice and the GFP distribution in 3 slices, where the background stain is for nu-N for neurons, GFAP for astrocytes, and IBA1 for microglia. And you can see the clear improvement in delivery on the FUS-targeted hemisphere, although it is somewhat heterogeneous over the area of disruption. And then if we zoom in to quantify the percent of cells transfected, we can see here that in both striatum and cortex, in areas where we saw high delivery and small ROIs with high delivery, we get about 25% of total neurons transfected. And then we also looked at the other cell types, astrocytes and microglia, and found that the ratio of transfected cells was roughly 15 neurons to 8 astrocytes to 4 microglia. So we looked at 2 different markers of microglia activation to see if we could detect any kind of inflammatory response. The first column is showing IBA1 staining, and you can see that in one of the mice we did see maybe a hint of some microglia activation in the striatum and cortex on the FUS-targeted side. When we looked at CD68 as a second marker, we did not see any evidence of inflammation. Similarly, for astrocytes, using both GFAP and vimentin as 2 different markers, again, we saw potentially some hints of slight activation in some of the mice, but nothing that was widespread. And it should also be emphasized that this was at a 3-week time point, so this is more likely due to the presence of the GFP or vector than the ultrasound BBB disruption. So this is the initial results from study 2, where we're looking at BBB disruption in these 4 different groups. So again, the disruption was targeted to the right striatum. We did T1 weighted contrast MRI, and we registered it to a mouse template and then segmented out a volume of both the left and right striatum. And we're showing the images here as percent difference from pre-contrast injection. And you can see that we got pretty consistent opening over each of the 6 mice in all 4 groups. And here's the quantification of that, where on the left plot, we're showing the average signal intensity as a measure of percent signal change. And you can see that, interestingly, the only significant difference was actually between 2 different strains of the HD model mice at the same age, and just barely significant if we look at the volume of the striatum that was disrupted. And also, interestingly, when we looked at the Q175 model of Huntington's disease at the young versus older time point, we see a trend towards larger BBB disruption, but this did not reach the level of significance. And so now we're ready to move on to the histology, which we hope to have soon to quantify the level of GFP delivery and see if we can correlate it also to the amount of BBB disruption that we saw. So to conclude, in our baseline study, we largely replicated what others have shown in delivery of AAV vectors, and getting about 25% of neurons transducted in high delivery areas, although the delivery was quite heterogeneous. And there are some signs possibly of an inflammatory response at 3 weeks post-treatment, but not major. And now as we're starting to deliver in a disease model mice, we do see that we get pretty good BBB disruption across the mice and can cover a large portion of the striatal volume. And we see this potential effect of due to the different strains and possibly an effect due to age, but we're going to look further into that, and we'll get the GFP results soon. So thanks very much to all of our collaborators and groups, and thank you for listening.